Hi everyone, my name is Naina Beishembiva, and I'm a student from Kyrgyzstan, a Central Asian country, and I am a currently rising senior studying at Lake Forest College and double majoring in chemistry and data science. This summer, I was able to work at Pacific Northwest National Laboratory with an amazing team under the mentorship of Brittany Gorman, Jeremy Clare, and Chris Anderton, working on a topic of enhancing spatial lipidomics of lung tissue through multi-mass spectrometry and immunofluorescence microscopy with advanced R coding. So coming to and exploring the background of why are we even studying lipidomics? Well, lipids play an important role as a part of the human body in general. And if we take a look at an example of our lung tissue, it can be subdivided into bronchi, bronchiole, alveoli, and in alveoli, the lipid tissues play an important role in its elasticity and therefore playing a major role in a gas exchange. And so we want to create a method to identify and characterize those different regions. And in this microscopy slide of a lung tissue, we can see that it's even further subdivided into alveoli, airway, and vessel. And each compartment of this tissue has a different molecular composition. And so coming back to our example with alveoli, the changes in cellular lipids can indicate changes related to tissue functioning. So for instance, if in alveoli, the lipidomic composition has been altered, it might affect our breathing, in other words. So one of the main techniques that we use for this research is called matrix-assisted laser desorption ionization mass spectrometry. And the way it works is that we have our lung tissue sample sprayed with ionic matrix, and then we apply our laser that shoots our sample and as a result creates this mass spectrum. So at x-axis of our spectrum, we have a molecular mass that's created, and then in y-axis, we have our intensity. And at every single peak of our mass spectrum, we sort of can create this ion map. And at every pixel of this ion map, every pixel of the ion map will represent where the laser has shot our sample. And so to describe in general our experimental workflow, we have our tissue that has been sectioned, a donor tissue that has been sectioned using cryostat. We then mount it into slides and we apply our ionic matrix and run it through multi-mass spectrometry imaging. However, my research for this summer was focused on the second line of experimental workflow, and I'll describe it more in detail. Once we ran our sample through multi-mass spectrometry and obtained all the spectra, we then upload it to Metaspace. And Metaspace is an open source online software that will identify and annotate molecules based on various databases. So here you can see an example of a lung data that has been run through a multi-mass spectrometry imaging and was uploaded into Metaspace, and the way it has been annotated. Once we annotate our data in Metaspace, we run the same sample that has been already analyzed through all these steps now at immunohistochemistry staining and imaging. And to describe in very short summary how immunohistochemistry works is that we have our tissue sample, and our tissue sample contains many various cells. And as an analogy here, I put different cells represented by different figures and colored in different colors. We then apply a primary antibody, which is very specific to certain cells or antigens. And in this instance, our primary antibody binds specifically to a circle colored in red. We then apply a secondary antibody, which will bind to our primary antibody, and our secondary antibody contains a substrate that will create some sort of a certain color pop at our microscopy imaging later. As a part of my summer research experience in a wet lab, I was able to try to create my own protocol and optimize it for a kidney tissue, and here are the following results that I have. So for the tissue functional units of my interest, uromodulin, pondacolixin, and aquaporin-2, you can see that each of them have their own color to represent them. And in future slides, for each color and each tissue functional unit, I'll refer to them as a channel. So moving on, once we stained our tissue to tag all the areas of the interest and image them, we then upload all those imagings into a software called HuBMAP, which you've already heard of. So here is an example of a QPath annotation for a lung tissue. And in this case, we have 22 channels to represent it. And here's an example of annotation of one of them for keratin 5. And each annotations were created by choosing specific immunofluorescence threshold, and it was done by hand. After we have finished annotating our images on QPath, we then upload this data into another software called Skills. And Skills is a Bruker mass spectrometry processing software, and it's very vital and important for us because it can combine both the data that we got from Metaspace with the mass to charge ratio values of our ionic maps, as well as the annotations imported from QPath all together at one place. So after that step is done, we then perform a statistical analysis in R to see what cells are more upregulated in specific regions of a tissue functional unit. Just to come back to a previous point and elaborate more on it, we have our segmentations in QPath where we segmented specific areas, and we combined it with our molecular annotations made in Metaspace together in Skills. And so by overlaying these two pieces of data, this is what we get. For each pixel of our image now, we will know the spatial coordinates of it, so the XY values, as well as ion intensity, or in other words, we will know what molecules potentially could it be, and we will know what segment does it belong to. Whoops. I think it skips a slide. And to dive more deeper into our R analysis or statistical analysis in R programming language, we first started by importing a library called Skills Lab Client, which is a Skills API function that helps us basically to bring all the data that we put up together at Skills software into our programming language, as well as using an aromics processor data, which has been created here at Pacific Northwest National Lab by a scientist called Jeremy Clare. And you can access it by following this QR code or this link, which will take you to a GitHub page. And this aromics processor data has many functions to analyze the omics data. We then put all the data together into a huge data frame using a function called aromics create object. And here is an example of two sets of data that I am trying to bring in together into a huge table containing our mass to charge ratio values with the intensity, as well as the XY coordinates and the channel annotations. We then perform on this huge table set logarithmic transformations and median centering, which helps us to normalize our data and perform further stats on it. After that step, we go on to perform two-tailed test, and two-tailed meaning that it will allow us to see either upregulation or downregulation of the molecules at a certain region. And it will compare a selected area or a tissue functional unit that we're interested in to the rest of the tissue in the statistical analysis. And once we have our results, we then want to visualize the results by creating a Violin plots. And so moving on to results, we started testing and validating this workflow using kidney data, since it only had 2 channels, and we were able to identify these specific lipids in these corresponding regions of uromodulin and megalin. And you can see our findings here supported by the Violin plots. However, we then moved on to our lung data, which contained 22 channels. And here you can see a result for 2 of them called Seline, which is a molecule which stands for an alveoli type 1 cells, and SFTPC, that stands for alveoli type 2 cells. And we were able to find these following molecules that are more upregulated at these corresponding regions called N-glycans. And the study of N-glycans is applicable to many fibrotic diseases that occur in alveoli. And to go more deeper in detail of these Violin plots, in blue, we see the region of our interest, such as Seline or as SFTPC regions. And we can see that their presence their presence is more upregulated than the presence of any other molecules than the presence of this molecule in any other region of our tissue. Sorry. And moving on to conclusions, this workflow enabled us to annotate lipids and glycans, and we were able to validate this information and workflow on kidney data as well. Going further, coding in R allowed us to then perform even a statistical comparison of the abundance of these ions within each region of interest. And our future applications and ongoing work right now is that we can apply this workflow to other human organs and other spatial assays such as N-glycomics and proteomics. And in future, developing more advanced coding tools can be used to compare healthy versus diseased tissues. And we are right now also working on new functions, developing more the Aromics library to allow to perform statistical analysis on functional units based on combining immunofluorescence labels such as alveoli together with Celian or SFTPC and others. And lastly, I want to thank again my mentors, Brittany, Jeremy, and Chris, for their continuous support and their kindness and big inspiration, as well as my entire lab, under 10 lab, and in general for this summer research, which I was able to do due to the help of NIH and HuBMAP, as well as thank you to Pacific Northwest National Lab. And thank you so much for your attention. Let me know if you have any questions.